This lesson is about translation, or how we take the information in DNA to build proteins. And translation is the second half of that process. Remember, the first half was called transcription, and that's where we use the information in DNA to build RNA. Now, in translation, we're going to use the information in RNA to build a protein. Let's just review the central idea or central dogma of molecular genetics. The main idea is that DNA is our genetic instructions, and it can make more DNA in a process called replication. Also, DNA can make RNA in a process called transcription. After RNA is manufactured in the nucleus of eukaryotic cells, it is often processed where the introns are cut out and the exons are spliced together, and that produces mRNA, and this process is called RNA processing or editing. The messenger RNA building proteins is called translation. And all of these processes regulating the final end product or trait that we receive is called expression, gene expression. Now let's look in detail at the process of translation. So this is a diagram of what would be in a eukaryote cell in the cytoplasm, and the messenger RNA is shown here attached to a ribosome. So let's just go ahead and label some of these things on this diagram. Ribosomes come in two pieces, the large ribosomal subunit and, you guessed it, the small ribosomal subunit. Sandwiched between them is the messenger RNA. The G' cap, the modified guanine, is there to help the messenger RNA in the right orientation so that it knows where the beginning is from the end. And actually the messenger RNA attaches to part of the ribosome first, slides through until it gets to the right position, and then the rest of the ribosome attaches and clamps down on it. And the ribosome is going to be serving as a kind of a work area to assemble the protein together. Think of it as a workbench. Floating around in the cytoplasm are also amino acids. Remember, there are 20 different kinds. There's thousands of them floating around. And also floating around are small molecules of RNA that are folded up in a particular shape because of the way the bases are arranged, so that they kind of represent, in a way, look like maybe upside down clover leaves. These are called transfer RNAs. Transfer RNA or tRNA. Notice that the messenger RNA I've arranged in sets of three, groups of three bases each. Each base is called a codon. Now the way that the process of translation works to gather the right amino acids together to build the protein, and not just any amino acids, which would just build a protein of junk, is that the codon of the messenger RNA matches up with a complementary anticodon on a transfer RNA. The anticodon is the bottom three bases of a transfer RNA, and they will match up with each codon. Now each transfer RNA, and there's 64 different kinds of transfer RNAs. Notice how each one of them is slightly different. The bottom bases, the anticodons, are all different. For example, one might have a UAC anticodon, another one GGA or UUC. Anyway, there's, with three letters, three bases each, there's 64 combinations. So it turns out there's 64 kinds of transfer RNAs. Now because there's only 20 kinds of amino acids, it turns out that different transfer RNAs can pick up and combine with these amino acids. Say, for example, the transfer RNA that has the anticodon UAC binds to, I don't know, pick your favorite amino acid. Very good, let's say it's the heart-shaped amino acid. It turns out that the transfer RNA that has the anticodon UAC will always bind to only that amino acid, only the heart. Always the heart, nothing but the heart. Maybe the one that has the anticodon GGA could bind to a different one, say the triangle. And the one that has a UUC could be a different one than that, maybe the lucky horseshoe. This idea that each different kind of transfer RNA picks up a different kind of amino acid. Say, for example, one that ends in UUA could also pick up the lucky horseshoe. Now, just because UUC anticodon transfer RNA picked up the horseshoe amino acid doesn't mean that another one can't pick up the same amino acid. What we mean is that the one that has UUC will always pick up the horseshoe and only the horseshoe. Also, the one that ends in UUA will always pick up the horseshoe. It could never pick up, say, the square or the circle or the rainbow, just the horseshoe. But because there's 64 kinds of transfer RNAs and only 20 kinds of amino acids, there's going to be some redundancy. So more than one kind of transfer RNA can transport the same kind of amino acid. And what they do is the transfer RNAs bind to the amino acids, become charged, we call it. Then they float over to the ribosome and fit in the ribosome where the anticodon will match up to the codon. They will temporarily hydrogen bond in place. So there's the first transfer RNA in the ribosome. There's room for a second transfer RNA. So the transfer RNA with the appropriate anticodon, which in this case would be GGA, moves into the ribosome and it takes the second spot, temporarily hydrogen bonding. So there they are, two amino acids on two transfer RNAs sitting in the ribosome, held in place, and they can be there long enough for a dehydration reaction to occur between them, binding those two amino acids together. Remember, the structure of a real amino acid looks like this. There's a central carbon atom with an amino group on the left, a carboxyl group on the right, an R group, which could be 20 different things. Now if we put a second amino acid next to that, there it is. And notice the hydroxyl group of the first amino acid can do a dehydration reaction with that second amino acid amino group, releasing a water molecule. Once the dehydration reaction occurs, the carbon and the nitrogen bond together, forming a peptide bond. It's just a covalent bond between a carbon and a nitrogen, but because it's in a protein, they give it a special name and call it a peptide bond. So that's how amino acids really bond together to begin forming a protein. The next step is that the hydrogen bonds from the first amino acid holding the first transfer RNA in place in the ribosome are broken, and the ribosome moves down, or you can think of it as the messenger RNA has slid through the ribosome. Because the hydrogen bonds are no longer holding it in place, and when the peptide bond forms, it allows the break between the transfer RNA and the protein to occur. Nothing is holding this transfer RNA in place, so it floats away and finds another amino acid, another heart-shaped amino acid to bind to. That leaves an open spot in the ribosome. Once the ribosome slides down, or the messenger RNA slides through it, carrying the second transfer RNA that's still hydrogen bound to the codon of messenger RNA. So we'll redraw the ribosome in a different position. Okay, that opens up a new spot in the ribosome for the next transfer RNA to come and bind. And of course, that one's going to have which anticodon? That's right, the complementary one would be UUC, which just happens to be carrying the horseshoe-shaped amino acid. It hydrogen bonds in place. That puts two amino acids, the triangle and the horseshoe, right close together. A dehydration reaction can occur, forming a second peptide bond. At that point, the bond to the triangle shaped amino acid and the transfer RNA break, and the hydrogen bonds are temporary, they break, and the second transfer RNA floats away. Again, the ribosome moves down, or the messenger RNA slides through the ribosome. But the amino acids stay linked together with these peptide bonds. And this process just continues over and over with new transfer RNAs fitting into the ribosome, new peptide bonds forming between the amino acids, until if there's ever a transfer RNA that doesn't have the correct anticodon, it just doesn't stick in place long enough for a peptide bond to form. And this way the amino acids are put together in the correct order and coded for in the instructions in the RNA, which was built from the DNA. Now this process continues until a codon is reached that binds up with a transfer RNA that does not carry any amino acids. When the transfer RNA that carries no amino acid binds to the messenger RNA, it acts as a release factor. With nothing for the previous amino acid to bind to, it releases the string of amino acids, which can now travel into the cytoplasm of the cell and do whatever function that they do. They float away. The ribosome falls apart, separating the large and small subunits, and the transfer RNA floats away as well. The transfer RNAs that code for no amino acids are called release factors, and the codons of the messenger RNA that code for them are called stop codons. In essence, they act as the signal to tell the protein when to stop being built. So at this point, now we have a completed protein. It can start folding and bending and twisting according to the rules for protein folding and be transported off to do its job in the cell. The instructions for that protein to get the amino acids in the correct order came from the order of bases in the DNA that built the RNA that was then used and translated into protein. And that's how the process of translation occurs.